vision of the group is really to make sure that the experiments are performed under conditions that are very close to natural conditions. And what we try to do is really work on biological systems, and we want to apply physical technologies and physical models in order to understand them. The Physical Biology Group is a member of the Department of Life Sciences on the campus Riedberg of the Goethe-University in Frankfurt am Main. The group actively links physics and biology. We employ people from various different areas: chemists, pharmacologists, biologists, biotechnologists, and of course also engineers, mathematicians, and physicists.
The idea of our work is that we are gathering as much detailed knowledge as possible on a diverse set of biological model organisms and biological model systems. And we are hoping in the end that based on this knowledge in future we are able to develop novel technologies that will actually bring society forward.
We should always look at cell communities. That is really the issue. If we work with an insect, for example, because I really think that is actually also a multicellular structure, we have to make sure that it really develops into a morphologically intact specimen. Otherwise, we cannot be sure that the small event that we are looking at is really resembling what happens in nature.
We apply physical methods, and particularly the light microscopy, fluorescence microscopy, to analyze our biological specimens in 3D. And the main tool for that we use and we actively developed is a light sheet fluorescence microscopy, or short LSFM.
Light sheet-based fluorescence microscopy is a very, very sample-friendly imaging method. Since the energy irradiation on the specimen is extremely low, we are able to preserve the three-dimensional morphology of our samples and thus gain novel insight which would not be possible to get with any other fluorescence imaging technique.
The most important feature of LSFM is exposure to minimal light, so that the specimens are preserved over a long period of time, alive and well. And the second feature is having very fast three-dimensional images of your specimen.
The three-dimensional structure of specimen is very important for us to understand, especially in the context of tissue engineering and 3D bioprinting, as we try to replicate these complex structures such as vasculature and stem cell niches. And therefore, after imaging high-resolution specimen, we focus on generating the microstructures that will accurately mimic the 3D environment. And to do so, we 3D print cells in a hydrogel with a technique that we call light sheet photopolymerization. And we're confident this is going to make a big difference in the field of 3D bioprinting.